Welcome student. In the last lecture of this week, in which we will be discussing about theories of organization of root apical meristem. In the previous lecture we have discussed about various theories that was for root apical meristem, in which we have also discussed that some of things are common for root apical meristem also, and in this lecture we will try to analyze those things in terms of root, as well as some new theories are also there which is only applicable for root apical meristem. So coming to the first theory, now in root apical meristem also scientists have studied so many different plant types, and they finally came that to the conclusion that there are two basically types of cells which are there in root apical meristem. In one case there is a single apical cell, so the root apical meristem is unicellular. Okay, a single apical cell is there which is found in ferns, whereas in other root of other higher plant types, the root apical meristem is multicellular. Okay, which is also found in some pteridophytes like in lycophytes and few other ferns also, few lower groups of plant types, or you can say vascular cryptogams, and it is also found in gymnosperm and angiosperm. So first talk about apical cell theory. This also we have discussed in shoot apical meristem, that according to this theory there is a single apical cell which give rise to all parts of the root as well as shoot. So in terms of root also, if we try to analyze the root apical meristem or the apical cell which is found in the root, we can see here that it is also a tetrahedral structure forming of 4 faces. Now in this case slightly it will be different because the position of shoot apical meristem is terminal, whereas root apical meristem is subterminal. Now in this case also we can see a single apical cell, okay, which is tetrahedral. The 3 sides are on the inner side of the root, whereas one side is found on the outer side of this root. Now the inner 3 sides give rise to segments which produces the body of the root, whereas the root apical initial on this side, okay, on the other side give rise to root cap. So in this case there is also a slight difference, because in shoot apical meristem the terminal cell which was lying on the top side was actually not contributing in formation of any of the cells, whereas in case of root apical meristem the one which is lying on the bottom side, it is contributing in the formation of root cap. Okay. The other side will form the epidermis, they will form the cortex, as well as they will form the vascular cylinder. Now the apical cell is partially vacuolated because it is also dividing, so vacuolation is not required as such, presence of vacuole, but yes, in older cells sometimes it has been observed that this vacuolation can increase. Now the histogen theory that also we have discussed, it is also applicable for root and shoot, and as I have told you before that it is more applicable for root. Okay. So this was proposed by Hanstein, and according to this theory there are basically three layers of tissue. Okay, and these three layers of tissue are known as dermatogen, peribellum and pleurome. So that also we have discussed before. Okay, so here also you will have three layers of cell. The lower layer will be known as dermatogen, and then you have peribellum, and then you have pleurome. Now pleurome will give rise to vascular tissue including pith, peribellum will give rise to the cortex, okay, and dermatogen will form the epidermis as well as the root cap. Okay, so this, but before also we have discussed that this theory, although it can explain the layering pattern and it can explain the development of different cell types from these three layers, but it was observed that it could not clearly define many of the structures, because in many of the plant types these layers cannot be distinguished and their functions could not be assigned, because according to histogen theory these layers are distinct. They have a particular destiny, they are committal and they are giving rise to a particular cell type. But in root also, as we have discussed before, that in roots you can find, when we have studied the organization of root apices, we found that there are different patterns. So, okay, apical cell theory is applicable for vascular cryptogams, but in other higher plants also it was observed that root was containing different types of these layers, means different numbers of these layers. Like you can have a plant type in which only one layer was there. Okay, so this one group of initials, they were giving rise to all different parts of the plant. They were also giving rise to root cap, they were also giving rise to epidermis, cortex and vascular tissue. Then in some cases you can have, it was observed that there are basically two layers. Okay, some layer is giving rise to root cap, the other is giving rise to other parts of the plant. In monocot particularly, it was observed that there is a fourth histogen also. Okay, in which the lower one was giving rise to root cap and the rest were behaving like dermatogen, peribellum, as well as pleuromes, giving rise to other different parts of this plant. So this theory was also not so supported, because in root also a lot of variation was there, although it could explain some plant types, but later on different theories have, as they have replaced in shoot case also, so many other things also came in case of root also. So the next important concept or the theory which was given in case of root, and it is particularly applicable only for root, is the Corper and Kappe theory. Corper means body and Kappe means cap. This theory was given by Scoop in 1970. So you can see here that all those theories are long back. People have been working for so long and they were studying for so, means long back also they have studied a lot and they have tried to understand the organization of these tissues. Now this theory is based on the difference in the plane of cell division. According to this theory, the root apex divide in a pattern called T division. Okay. Now, if we try to understand this, see in root tissues, lot of cells are there. Okay. these cells are there. Now in this, suppose this is a particular cell, okay. So in this case the first division will be transverse, okay. Transverse means in this plane, okay. Suppose this is the cell, this is the transverse division. Now the next division will occur in the same cell. See, two types of cells have been formed. In this, one cell will again become active and it will divide longitudinally. You can say it will divide longitudinally. Now if this type of division occurs, you can see it is forming a T-shape pattern, okay. So most of the cells, they will divide in this T-shape pattern. Another type of division can happen if suppose this is the cell and it has divided by a transverse division and the above cell divides longitudinally. So you can say that it will have a inverted T-shape pattern, okay. Now the outer regions of this, when we analyze this root tip, we can say the outer regions are actually known as, they are known as Kappa, which divides by this, by a pattern which is known as Kappa pattern, okay. And the middle one, they divide by Korper pattern, that is the forms the body. Now in the outer region you have a T-shaped structure which is the cap-like pattern, okay. And in the inner regions, okay, in the inner regions of this root apex you will have Korper type of pattern which is the inverted T type, okay. Now, in this case, we can see here that when we observe the meristematic region, we can see that they are made up of multicellular and contains longitudinal files of cells. You can see a lot of longitudinal cells are there, and when the cell is growing and it is dividing, it happens that they divide in such a fashion, they divide in a T shape, like one cell transverse division and the other cell longitudinal division. So, in this case you can see here that this is a single cell, suppose it has divided, the other one, the outer one has divided, forming a T-shaped structure. Suppose they are forming a T-shaped structure. So, in that case, the cells will actually increase, they will actually also slightly try to spread out. So, in this way, the division will occur in the root zone, the cell number will increase, and they will, a single longitudinal cell will become double, and both out of two cells, one cell will retain the capability of division and it will give rise to two cells, and again both the daughter cells will inherit the capability of division. Now, both the daughter cells will be actually in parallel orientation and they will share a common wall and will divide again by transverse division. Again, one of them will again divide by longitudinal division. So, in this way you can find lot of files of longitudinally elongated cell, and in the columella region you can see here the inverted T-shaped structure. See, you can see here this is inverted, whereas in the lateral root cap region you can see a normal T-shaped structure where the cells have divided in this fashion. So, this region, the columella region, is known as corpor, forms the body of this root, and this forms the cap. Okay. The T division in this region is the Kappe region, which is also known as the cap region. Similarly, in the above they will divide, and these cells will finally be destined to form the cortex, epidermis and the stele. So, this is how the organization it is explained by Corpor and Kappe theory. Now, the next one is the quiescent center concept. See, as in shoot apical meristem also we have seen a central zone which was basically a group of cells which were not dividing, and their growth or the division index was much lower as compared to the other cell. In the same way, in root zone also we have quiescent center. Okay. So, according to this concept, there is this is the meristematic tissue. Okay. In the center of this meristematic tissue, you have quiescent center. Okay. The structure of this quiescent center is it is basically hemispherical, sometimes it is discoid in shape, and the number of cells in the quiescent center can vary. As less as 4 cells have been observed in Arabidopsis, and more than 1,000 cells are supposed to be present in quiescent center. And this is the place which serves to renew the more actively dividing regions which are present around the quiescent center. So, this quiescent center we have also analyzed when we were discussing about the structure of root apical meristem. In that case also we observed that there were initials which were arranged on either side of the quiescent center, and these initials were giving rise to different parts of the root. Okay. And this quiescent center, suppose if a root is broken, roots are prone to be broken many a times because they are growing inside the soil, so they are getting damaged with the course of their development. So, these initials or the other meristematic cells can be damaged. So, in that case the quiescent center can renew those cells. They could be damaged by many other environmental factors also, like presence of biotic flora or presence of the soil texture. So, all those things can damage those cells. Now, see this quiescent center, when the scientists have tried to analyze this quiescent center, they found that we cannot actually distinguish them on the basis of cell structure, because this is quite similar with the cells which are present on the meristematic region. So, how actually they identified? See, various different methods, like they fed the root apices with radio labeled chemicals, okay, and they tried to analyze the DNA synthesis. So, when they actually fed those cells, they found that the cells in this particular region were not dividing, whereas radioactivity was observed in the adjacent cells, whereas in the quiescent center the cells were inactive and they were not dividing, so no radioactivity was observed. So, on that basis they came to understand that these cells are non-dividing cells, and similar type, as I have told you before, that similar type of zone is also there in shoot apical meristem, which is known as central zone. Okay. So, this is all about the organization of root apical meristem and the theories which are developed by various group of scientists to make them understand how this root apical meristem has been organized. We have understood the meristematic tissues, we have understood what are the different types of meristematic tissue and where they are organized. Now, next, in the next class we will move on to permanent tissues, that means the tissues which have been differentiated and which have been formed from the meristematic tissue to perform a specific function, and they have attained a specific structure.